Hello, my name is Harald Hunsberger. I'm going to show you our laboratory today. So the laboratory building is integrated into this beautiful campus. The area Krems is a biotech area. We find different universities here. We have companies in research and manufacturing in the biomedical area. And in the next minutes, I will show you the laboratories and all the instruments which you will face during your studies. Almost 50% of all the lectures and teaching units are done in the laboratory. So what we believe is that technical experience is highly important in the field of biotechnology. Therefore, we have modern laboratories, we have professors from different areas from science and industry. Let's see how our guest professors teach our students in modern equipped laboratories.
I come from Szeboń. I work there at the Institute of the Botany from Academy of Sciences. And here I'm working as an external lecturer. I'm teaching the biochemistry laboratory and also this genetic engineering.
I'm Yasmin Mamnon. I'm an external lecturer here. I'm teaching cell culture techniques. I am from the biopharmaceutical industry. I like coming here every year to teach the students because I think it's very important that the students get input from very different fields, not only the academic here, but also from biopharmaceutical companies to see what they can expect when they go to industry.
One of our major focus points in our curricula in the biotech area is internationalization. So first of all, our students come from different countries. Another example in our bachelor degree programs are the exchange semesters in the third and fourth semester. And last but not least, a very attractive opportunity to do the bachelor thesis is of course the practical training semester. We had almost 200 students in the United States. Very attractive placements, where the latest technologies such as induced pluripotent stem cell technologies are being performed. Research is a very important aspect of all of our degree programs. The results we achieve in our research projects are integrated into our curricula. One example is a PhD collaboration in the field of cancer research. We are analyzing with our PhD students therapy resistance of cancer cells.
Hello, my name is Alexandra Zalzalko and I'm doing my PhD here at the IMC Krems. In my PhD project, I focus on the investigation of the AXL receptor tyrosine kinase, which is overexpressed in many cancers and leads to the therapy resistance. What I want to do in my project is to produce a specific inhibitor against AXL. For that, I will use yeast cells, and the goal is to identify a novel therapy strategy for cancer patients.
Hello, my name is Florian Eibensteiner and I'm also working on the topic project with Alexandra. We cooperate with the molecular pathology in St. Pölten, where we get our clinical samples from. I'm investigating how AXL forms the therapy resistance in cancer patients.
My name is Christoph Metzner and I am the current Program Director of the Master's Program Medical and Pharmaceutical Biotechnology, and I'd like to show you some of the bits and pieces that are special to our program. We have a very diverse range of research projects that are tightly associated with the program, and most of them, what they have in common, that most of them start here within the cell culture labs. What we do here is preparing samples that we get, for example, from patients, but also we work a lot on cultured cells, where we try to start experiments where we treat them with potential therapy methods to test in the cell culture setting.
Hello, my name is Andreas Lackner. I'm the Program Director of the Omics Technologies and Data Science in Biomedicine Master Program. This is a part-time master program, so it enables students to work and study at the same time. And we particularly focus on a set of technologies that's called omics technologies that include genomics, transcriptomics, proteomics, and metabolomics. We provide our students the means to analyze the huge amount of data emerging from these technologies and then apply them to questions in personalized medicine and diagnostics of the future.
Most of our experiments in research, as well as a lot of the teaching, starts actually here within the cell culture labs. We work a lot with these biosafety cabinets, what's colloquially known as a laminar hood. This is intended to protect the environment as well as your samples, and of course also the operator from any biocontamination or biohazard, in order to be able to work safely with cell cultures or patient samples. When we initiate our research projects, we usually do this on the cell culture level, as we've seen previously. So we work on a small scale level. What is very important when we want to do the translation into the more industrial related areas, pharmaceutical or biotech, we have to go for larger volumes and use completely different systems. This is mostly done in bioreactors. That, of course, is not the size you would see in the pharmaceutical industry, but the level of control and the level of issues that we see are comparable to what you would find in pharmaceutical industries.
Our programs, the bachelor and the master degrees, are centered around the development of modern therapies, such as monoclonal antibodies for cancer treatment. So all these therapeutic proteins are manufactured in bioreactors.
My name is Hans-Peter Weizenberg. I'm here in the MassSpec facility where we also do applied research. So the IMC is also about collaborations with companies. For example, one of our research projects is about characterizing bone allografts for tissue engineering with one of our collaborators, where we also identify the proteins in those allografts with our setup. Another speciality is also our sister institute, which is concerned with also omics technologies like the MassSpec. We have a stronger upgraded device version over there, which is mainly used in industry corporations, but also integrated into the study programs. For the more challenging objectives in, for example, the omics study program, we can also access this one. And our students also have a chance to do their practica on this device.
This is our DNA sequencing machine, the AVITI. With this machine, we can analyze the entire DNA content of a cell or the entire RNA content of a cell. Meaning we can really analyze all the small differences in DNA that emerge either in individuals or in diseased versus healthy cells. Procedures like this are used in research to find more about the systems level of cellular mechanisms and can be used in clinics to tailor individual therapies for patients. DNA sequencing is heavily used in current research questions. To tackle these current research questions, we also have a state-of-the-art laboratory in the building where we can do tissue culture and then look at our cells with this confocal microscope, for example, where we can really section the cell layers and look at very tiny structures within the cells that are fluorescently labeled to make them visible. This is the end of our tour. Thanks so much for joining and see you at the IMC campus.